morning I'm going to talk to you about a non-AFM but type of scan probe microscopy, and it's electrochemical microscopy. And I'm going to really introduce the instrument to you more than really digging down into the applications, but I'm going to give a few examples at the end of this presentation. So not all of us here know electrochemistry. I know a battery section, many people do, but I thought I'd start off for the general AFM audience. Electrochemistry is really the fusion of electricity and chemistry. So we've got electrons in a wire side of a system, exchanging electrons either with molecules or having ions insertion in a chemical system, and kind of the governing law that we think about in this process is Faraday's law, and that relates the current in the wire to the rate of whatever the chemical process is on the surface, and that underlines any electrochemical process, be it batteries or many other things. Electrochemical science appears in tons of technology. Probably the biggest market is thinking about corrosion is at the heart of it, an electrochemical process, but there are electrochemical sensing, thinking about glucose sensing, perhaps the biggest one that we're familiar with. Batteries, you're going to hear a whole bunch about in the future in the rest of this session. But electrochemical synthesis of both bulk chemicals and fine chemicals, electroplating, fuel cells, many more that I don't have a chance to go into today, but this kind of microscopy I'm going to present could be interesting to people in all of these fields. So kind of a motivation for doing nanoscale electrochemistry. Well, what does normal electrochemistry look like? And what a normal electrochemical experiment looks like is typically has 2 or maybe more electrodes, could be immersed in a solution or separated by an electrolyte in a battery. We connect up a power supply, and we're measuring the current. Something's frozen.
Martin, we can hear you.
Okay, you can hear me. Okay,
yes.
That's the stuff. Well, while my slides are frozen, I'll carry on talking. And so in this measurement, we're going to measure, we're going to either control the potential or the current, and we're going to measure whatever that other variable is. We're going to, there we go, we started moving again. And we'll plot that in a bunch of different ways, depending on the electrochemical technique. As say, the most common example would be plotting a current versus a voltage response. In electrochemistry, we call that a cyclic voltammogram. And the currents will depend on the size of the electrode and such like, but might be on the order of a milliamp or something like that. And as I said, we're measuring the current, but we can relate that to a reaction rate over the whole surface of the electrode. But that's really equivalent to measuring an average rate over. So if we just divide through by the area, we're learning something average about a chemical process. But the real electrodes are never just one thing. If we're thinking, and this is shamelessly pulled from the literature, but a kind of schematic of what an SEI and a battery anode might look like, these are more likely to be used in electrochemical synthesis or fuel cells. They're nanoparticles on a carbon surface. We can think even a pure electrode has some structure to it in terms of steps and default. 2-dimensional materials, we've just had a whole talk on, so I don't need to mention anything about them. And you'll have to trust me, I'll show you this later. The electrochemical behavior of each and every part of this electrode can be different. And so taking one average measurement, a single value for the whole surface, is just averaging all of those processes out. And maybe I don't need to be laboring this to a scan probe audience. We all think about looking at the detail of whatever process we're looking at. And so really a traditional electrochemical experiment doesn't do that, but moving to the scan probe regime does. And so the goal, like with many scan probe measurements, is really to shrink the region we measure. And in this case, that to me is going to mean shrinking the area of the electrode that's immersed in the solution. And you will see a few other solutions to this problem later today, and unfortunately I don't have a chance to go into them. And the concept is shown on the left and is really rather simple. So what we're going to do is take a very small pipette and fill it with the same solution that we would use in the regular large experiment. Apologies, I didn't color them in the same way. And we touch it to the surface of our electrode. And the key is the end of the pipette is on the order of 100 nanometers, and surface tension is going to keep that droplet area being approximately the same size as the end of the pipette. And everything else is the same. If you look, we're controlling the voltage in both cases, measuring the current. Okay, I've labeled it slightly differently. Still got the same processes going on. We've got this area acts as the same as this whole electrode, and then this wire stuck in there acts as the other electrode. And really nothing else is different. And so this, to an electrochemist, is just a very weird shaped cell, but it allows people to do the same electrochemical characterization they've always been doing. But the difference is this order of 100 nanometer sort of radius cell is going to give something like 10 to the minus 14 meters squared area versus your typical perhaps 1 centimeter squared sample giving 10 to the minus 4. So and these are sort of rough orders of magnitude. So how do we make such a probe? Well, those of you that do SICM would know. We simply take a glass tube, a glass capillary, we heat it, and we pull it. And there are instruments to do this. I can teach a high school student to do this in about 5 minutes, and they're as good as I am. It costs $1 a time and a minute. Okay, the instrument costs somewhat more, but it's super simple. And kind of, you can see them either optically, you can get an idea, but if you look at the end of them, you can pull these to a few tens of nanometers or get a better cross section of them in a TEM. And long story short, I wouldn't be here. The technique works. We get stable electrochemical measurements. Here is for just simple electrochemical system. For those of you that want the chemical name, or it's written down there. The currents are a lot smaller than the milliamps I was looking earlier. We're talking picoamps or so, but these are measurable. And as long as we put a Faraday cage around the instrument, we can measure these above noise. There's some theory curves in here, and I shan't go into that today, but to interpret the measurements and to understand what's going on, we use sort of typical electrochemical models, and in this case, the green curve fits better than the red curve. We're in a scan probe conference, so it's perhaps no surprise to you, we don't just do one measurement of 100 nanometer area, but we do a bunch of measurements. And our operating procedure is, we call it hopping mode, but it's pretty similar to doing, say, force distance curves at every point. We touch the tip to the sample, pick it up, and we're doing a point-by-point measurement. We're still controlling the sample. The data set we get, we kind of at each XY position, we're going to have the current versus the potential. And so, again, you AFM people, think of an analogy to a force volume kind of measurement. We've just got an extra dimension. The way we quite like to visualize the data is through what we call electrochemical movie. You're about to see that, and that's a series of image plots where the color represents the current, and we're going to be gradually changing the voltage and seeing basically which parts of the sample light up, and those are the parts with the high electrochemical current. And this example is nanoparticles embedded in a Nafion film with a carbon support underneath. And for those of you that are familiar with fuel cells, this is exactly a model to the fuel cells. And so if we look at this image, you don't have to be an electrochemical expert. That's kind of what's nice about this technique. I hope all of you can tell where the sample is more active and where it's less active. So the currents are plotted as negative, and the green areas are really those high activity areas. So how sort of functionally do we get this tip down to the surface? Well, as with most scan probe instruments, we're going to be using piezos to do our fine control. And we've got this electrical circuit. We've got one electrode inside the pipette in the solution, and the other electrode, we're electrically connecting up our sample. But if you notice, we've got a gap in the circuit. And so high school physics tells us if we don't complete the circuit, we get no current. And so we've got to think what happens as we move the tip towards the surface. Well, I'm going to show you up here real data of what happens. We get this is current on the y-axis and position on the x-axis. We move the tip down, and the second we complete the circuit, we get a current. And the instrument stops the pipette approaching the surface more. If we didn't, that glass is very fragile and it wouldn't end up very well for our probe. It would shatter all over our surface. And once we're there, we perform basically whatever electrochemical characterization we choose. This one happens to be the kind of most common and quite a versatile electrochemical technique, but in principle, we can do whatever technique we choose. What does the instrument look like? Not too dissimilar to your to a typical AFM, perhaps. The probe holder has to be modified because we've got a pipette and we want to connect some wires to it. But that's a little bit different. We'll have some current amplifiers in the system, and then we've got the same kind of acoustic isolation. We'll want a Faraday cage, as I said, those low currents. And for our homemade one, we use an FPGA card to do some control of the system. And practically, it looks like this. I've got a sample scanner at the bottom and the Z scanner vertically. I put a camera in the side just to help me position things at the beginning, and some micro positioners for coarse approximation. That's really where the tip goes. And as you've seen before, it's, I say 100 nanometers, and that can be from a few tens of nanometers really up to maybe a micron in size, depending on what resolution, what area I want to scan. There are now commercially available systems. So this is a Park NX12 in a lab when I was in Utah. We got that put inside, well, I say we, and this is really thanks to all the Park engineers in Korea who engineered it so the vibrations weren't an issue, but inside an M Brown glove box. And so for those of you thinking, is this technique compatible with batteries? Well, if they're air sensitive and moisture sensitive, sure, you need to control the environment, but that's an engineering problem, and thankfully I didn't have to solve it myself. So finally, kind of, I'd love to talk to each of you and in the discussion session more about typical examples, but I wanted to really make sure you guys sort of saw how simple the technique is to perform, and then to just think and dream about how this would fit in each of your research. So I just pulled sort of some energy type applications from the literature so the first one, and this isn't my work, this is my former PhD group, but I think gives you a nice idea, perhaps might be interesting to some of you guys, is characterizing single cathode materials. So the concept is shown here. We're going to have a pipette filled with a battery electrolyte, maybe a propylene carbonate or something like that, with lithium ions in it. And we are going to put cathode particles on an inert support and spread them out quite far apart. And so we can touch the pipette tip to just one particle and characterize intercalation and deinsertion of lithium ions. Practically, this is an area that was imaged. You perhaps can't see the the spots perfectly, but each of those spots on there is a separate image. And the bright parts are the particles. So this is an SEM and it's a carbon support for those of you that care, but I encourage you to go ahead and look at the reference for the real details. These are just the currents taken at a particular potential. And so we can see that the bright spots do relate to the particles. And you can maybe see the merit of doing this measurement. The different particles have different electrochemical responses. There are bright ones and dark ones. Okay, well, that justifies us doing those kind of measurements, but we've got the SEM, so we can really look at each of these particles and we can do individual electrochemical characterizations at each cathode particle and really understand that there's some variability in the process here. You maybe have to stare a little bit at these diagrams. I apologize, they're shrunk, but the Y scale is certainly different. We're getting quite a few more ions into this particle. There's also a shift in the potential of when we can do that. And so rather than taking this lumped average approach, we can really sort of break this down to more fundamental scientific questions and characterization. So that would be a kind of reductionist approach for looking at battery materials. One can also look at them in a more in situ kind of fabricated with all the binder and the conductive materials, etc.
You have 3 minutes left.
Wonderful. I'm perfectly on time. Good. If we look in the bottom left, we've got, apologies again, shrinking all the diagram. We have a polycrystalline gold substrate here. So it's a pure metal, but the crystal grains are oriented in different orientations. And the schematic of just a pseudo-colored kind of thing is we're going to scan this pipette point by point over a surface. And here we're in a CO2 atmosphere. And I don't need to tell you guys why CO2 reduction is important, but having good catalysts for it and being able to electroreduce it really is. Here we've got a small section of an image over that boundary. The boundary, you can probably guess I'm going to tell you, is here. And what do we see? We see a higher current over the boundary, which tells you really the defects in the material are what are driving the reaction. And that, it turns out, it's to do with the strain in the crystal lattice. And there's a follow-up paper on that one. As the movie I showed you, and I've got a still from it here, is of platinum particles buried in a Nafion film, and that's a proton conducting film, the kind of thing you use in fuel cells. And like I say, I don't have time to drill down into any of these examples, but hopefully some of you guys are thinking, wait a minute, this might be the ideal tool to look at certain things in my research. And thankfully, the interpretation is simple. It is literally electrochemistry on a small area, and allows you to do these wonderful measurements. And of course, it wouldn't have been possible without the support. The project started out funded by the British Engineering and Physical Research Councils. You may have got my accent that I'm a European originally, but has followed on being supported by the Department of Energy, the Navy, the Air Force here in the USA. Thank Park, obviously, for the invitation, but also for loaning instruments and letting us work with you guys to get this technique on board with your instruments and the startup funds and the support here at the University of Arkansas. I got here in the middle of the pandemic last year, but fortunately have some students building up these instruments. And I guess I probably have time for one or two questions maybe, or are we holding those till the end?